We should start. Good afternoon. I'm János Szebeni. Yes, this should be heard. Yeah, it's on, I guess. I would like to open this afternoon seminar on toxicology of nanomedicines, and would like to call the first speaker Dr. Helinor Johnston from the Nano-Safety group at Heriot-Watt University in Edinburgh.
Can you hear me okay? Yeah? Okay. So the title of my presentation is investigating the relationship between nanomaterial behavior and their physical-chemical properties. So I just want to start by saying that typically we don't really work with nanomedicines in our group, but kind of the principles of the work that we do and the lessons that we learn are applicable to the nanomedicine community. So I'm sure you're all aware that nanomaterials that are exploited as nanomedicines are a really diverse group of substances. So they have the commonality that they're all very small in size, but they can vary with regards to a number of different physical-chemical properties. And so frequently, in parallel to the hazard investigations that we conduct, we try and characterize the physical-chemical properties of our nanomaterials. And this is to try and identify any attributes of these materials that are responsible for dictating their behavior. So controlling where they distribute to in the body, what controls their cellular uptake, their hazard, and their efficacy. We also tend to want to validate the information we've been provided with by commercial suppliers, just to check that it's correct information. And then we want to understand how the properties of our materials change when we suspend them in a biological medium. There's kind of a constant debate about which properties of nanomaterials should be characterized in parallel to our hazard investigations. And typically it's size, surface area charge, agglomeration, aggregation, and morphology that's prioritized, but there's a number of other aspects of the materials that you can also consider. Ideally as well, we tend to work with the pristine nanomaterials, and by this I mean what we've been supplied with, is it a powder or a suspension, but also nanomaterials that are dispersed within a physiologically relevant media, so for example cell culture medium used within in vitro experiments. There's a number of different techniques that can be used to characterize our nanomaterials, as I'm sure you're all aware. For example, for size, you could use electron microscopy, DLS, AFM, or XRD. The list is kind of endless. And quite often, investigators use what is available to them within their lab, for example, the equipment they have access to, the cost of performing that analysis, the time it takes to do this, and the expertise of the group. So it's a very multidisciplinary approach that's required, and often we use more than one approach to try and verify your findings, and we often get different results from different pieces of equipment. And importantly, not all the approaches are applicable to all forms of nanomaterials. For example, DLS isn't really suited to carbon nanotubes, it's more suited to spherical structures, and BET used to look at surface areas only applicable to powder. So we're limited in what we can do with some of these techniques. However, we really need to have a better understanding of how the physical-chemical properties of nanomaterials impact on their behavior, because in the future this is allow us to design more intelligently the safety of materials. So we try and manipulate the properties of the nanomaterials to improve, for example, their targeting or reduce their hazard. And it's also important for risk management as well and developing appropriate regulation. So I can't go through kind of the impact of all the different properties of nanomaterials on their behavior, but needless to say, there's been a large number of studies that have tried to investigate the relationship between the physical-chemical properties of nanomaterials and their behavior in the body. So there's kind of two main approaches that are used by investigators to do this. So the first one is kind of a hypothesis-driven approach. So the investigators have an idea of the property that's responsible for controlling nanomaterial toxicity and try and obtain a group of that type of nanomaterial that varies with regard to a specific property. An example that I'll go through today is the impact of carbon nanotube morphology on their hazard potential. More often than not, nowadays, it's more of a screening of nanomaterial toxicity that's performed. So we're given quite a large number of different types of nanomaterials that might vary with regards to their composition, their size, their morphology, and we screen their toxicity and compare how hazardous they are and try and identify what properties are controlling their behaviour. So as a first example with regards to how we would look at a kind of hypothesis testing approach, we'll look at carbon nanotubes, so kind of fibre-like structures that have a structural resemblance to asbestos fibres. So for asbestos is a structure-activity relationship that has been in existence for a number of years now that is able to predict the pathogenicity of fibres within the lung. So following exposure and deposition with the lung, a short fibre, and by this I mean something typically less than 10 microns in length, can be cleared by macrophages within the lung and removed. However, longer fibres and greater than 10 microns, if they're biopersistent in nature, macrophages clear to phagocytose them, leading to a persistent inflammatory and oxidative response which can cause damage. So it's this frustrated phagocytosis that's responsible quite often for the pathogenicity of these longer fibres. So we had a hypothesis that the morphology, and in particular the length of carbon nanotubes, is critical to their toxicity, and that the longer the carbon nanotube was, the greater its toxicity would be. So we wanted to identify if this fibre pathogenicity paradigm was applicable to nanotubes, and we used an in vitro macrophage model to test this hypothesis. So we had three types of nanotube: a long, straight fibre, which was about 50 microns, I think, in length. We had a long, straight fibre, which you can see here, but it was slightly entangled. And then we had one which we kind of refer to as our spaghetti. It was very entangled. You couldn't kind of see a long, straight fibre-like structure within that. We looked at the uptake just using light microscopy, and you can see the long, thin fibres were difficult to clear by the macrophages. They're kind of stretched along the length of the structure, whereas the kind of more entangled structures were more easily taken up by cells. This was also reflected in kind of the oxidative-based response and the inflammatory response, in that the longer the fibre was, the more likely it was to stimulate this frustrated phagocytosis, the release of reactive oxygen species and TNF-alpha from the macrophage cells. So this work demonstrated that the toxicity of carbon nanotubes did in fact conform to the fibre pathogenicity paradigm. This has been confirmed in Vivo studies conducted by our collaborators in Edinburgh. In terms of recommendations moving forward, you can use this to inform the safe design of carbon nanotubes in the future. And it's important to show that existing knowledge of particle and fibre toxicology can be useful within kind of newer materials. Obviously, it'd be good to have more studies that can take nanomaterials that have been systematically altered with regards to one property and try and investigate their toxicity, but it's very difficult to obtain these types of materials. Okay, the second study I'd like to discuss with you is a project called NanoMySex. This is an FP7-funded project looking at the toxicity of nanomaterials contained in inks. And the idea is that we identify which particles are most hazardous and then try to modify their surface to reduce their toxicity. So in terms of the particle panel, it was silver and aluminium oxide, a quantum dot, iron oxide, titanium dioxide, and zinc oxide. So here you can see kind of why they're used in pigments, the colours that they have. We used mammalian toxicology tests, both in vitro and in vivo models. And this is the results from the in vitro screening. So we used a macrophage cell, a lung epithelial, and a hepatocyte. And it was consistently the case that it was the zinc oxide particles that they're most toxic. So this is the LC50 values within the experiments, followed by the quantum dots and the silver. So these were always the most toxic particles, regardless of the cell type that we used. And it was actually the macrophages that appeared to be more sensitive to the toxicity. This finding was also confirmed within in vivo studies, where the silver and the zinc exhibited toxicity indicated by an infiltration of neutrophils into the lung. The quantum dots didn't in fact really have as much of an effect here. Just quickly as well, we did compare the toxicity of these nanomaterials using environmental models. So here we use the Daphnia, it's a water flea found in the aquatic environment, a worm which can inhabit water or sediment, and algae which inhabits the water. And what we found was that the ranking of toxicity of these materials was the same across the environmental and human models. So within this table, the red indicates that something's of high toxicity, the green of low toxicity, and the orange and yellow of kind of intermediate toxicity. So you can automatically see that across the different environmental models and the mammalian cell lines, that's the silver, the cadmium, and the zinc that is consistently toxic. So in terms of the conclusions from the study, the toxicity of the silver and the zinc might derive from the dissolution of ions. This is still being tested within the characterization. The macrophages were the most susceptible cell type, and the lumbriculus, the worm, the least sensitive. So we would recommend perhaps a tiered toxicology testing, that you might start within a macrophage model and maybe the Daphnia or the algae within the environment, and kind of screen the toxicity of your particles, and then look to more diverse models and to do your more mechanistic studies and a more in-depth investigation of toxicity. The next steps are to modify the particles, so they're going to take the zinc, the silver, and the quantum dots and try to functionalize our surface. We've received some that have been modified with some BSAs attached to the surface, and this has proved quite successful in reducing the toxicity within the pilot studies that have been conducted. So I know I'm kind of short on time, but lastly, I just want to discuss some of the work we've been doing to make in vitro models that we use slightly more relevant to the in vivo situation. So following exposure, nanomaterials become quite widely distributed. So this has been shown in a number of different studies. If we were to use in vivo models to test the toxicity of nanomaterials, it would cost a lot of money. Ethically, it wouldn't really be acceptable. So we need better in vitro models to screen the toxicity of nanomaterials at different target sites. During the transit of nanomaterials throughout the body, they tend to become coated with proteins, and this means that their physico-chemical properties change. So I think we heard yesterday quite a lot about the protein corona and the opsonization of nanomaterials. So what we suggest is that you kind of modify, these modifications that occur to the nanomaterials can be replicated within your in vitro models that you use. So you can better reflect the route of exposure and the biodistribution of your nanomaterials within the in vitro experiment. So the way that we have, this is just a schematic to show this. So for inhalation, the green on this diagram here represents lung lining fluid within the lung. And the idea is that the particles become coated with the proteins and lipids contained within this fluid, and then following the transit into the blood, you might get different serum proteins attaching. So it will depend on the affinity of these particles for the proteins for the particle surface, but also the physico-chemical properties of that nanomaterial will dictate which proteins bind. So we've done a few studies recently that have compared the toxicity of particles that have been uncoated, so kind of their naked, pristine form, versus those which have been coated with lung lining fluid, serum, or a combination of both, to see if this impacts on their toxicity. And we've looked at a variety of different cell types, again, hepatocytes, lung epithelial cells, and macrophages, and it seems that it is the kind of naked form of the particle that's consistently more toxic than those that have been coated with protein. So in conclusion, I'm sure you're all aware that the physico-chemical properties of nanomaterials impact on their behaviour, and the need to identify which properties make these materials hazardous. So we try, when possible, to characterise the properties of our nanomaterials in parallel to the hazard studies, and use any historical information available on particle and fibre toxicology to try and guide our hypotheses or help understand our results. this information can be used to improve the design of nanomaterials in the future or design appropriate legislation. As the exposure route influences the biological behaviour of nanomaterials by impacting on their physico-chemical properties, so this coating with proteins, for example, and you can reflect this within the design of the in vitro experiments. So as a few acknowledgements, I'd just like to thank different members of the Nano-Safety group at Heriot-Watt, in particular Vicky Stone and David Brown, who did a lot of this work, and I'll be happy to take, I don't know if questions happen now or later. But yes, thank you very much.
Thank you very much, Dr. Johnston. Questions, please?
Just a quick question. The higher sensitivity of the macrophages, does it correlate with increased uptake?
They didn't look at the uptake within that study, but I imagine that it would, that they were taking up the particles.
Let me ask then, if there is no other question. You were talking about physico-chemical properties to be found and identified as a potentially toxic. In your experience, what were those factors which you found as potentially toxic?
So across different projects that we've worked with, for example, silver has consistently been regarded as quite a high toxicology particle, and sometimes it's due to its solubility in the release of ions, and I think it's just its inherent toxicity of that material. It depends on the particle type, I guess. You know, morphology will be important, the fibers and carbon nanotubes, but for other size, surface area, are all important as well.
Well, if there is no further questions, then I'll have to